Now, okay, right. Thanks so much to the organizers of the Breakthrough Prize and to UCSF for putting on such a stimulating day of biology and physics and even math. We haven't talked about cancer today. Here we're going to talk about the future of cancer therapy. And this is a huge area of inquiry. Obviously, it's about a third of all pharmaceutical industry investment, big portion of the NIH budget. And we're really lucky tonight to have some real leaders in the field with complementary perspectives on where we're at and where we're going. So to my immediate right, Bob Weinberg. He's a professor at MIT and the Whitehead Institute, really one of the most influential cancer biologists of the past 50 years, Breakthrough Prize winner from 2013. Next to him, Napoleone Ferrara, another Breakthrough Prize winner from 2013 for his work back at Genentech, late 80s and throughout the 1990s on understanding the VEGF receptor and signaling pathway, vascular endothelial growth factor, which is a technical way of saying it's a form of how tumors can lay down blood vessels to grow and spread. And he figured out the way to make an antibody that would bind with that and curb that process. It's one of the most successful cancer drugs in the world today. Huge achievement. And on the far end, Michelle Arkin. She's a professor of pharmaceutical sciences here at UCSF, also has some biotech experience in her past, working on all kinds of new small molecules and different types of molecules that can bind with a lot of the targets that so many biologists have been working to describe over the years, new targets for cancer drugs. So now, for those who are not that familiar with the cancer story, it's kind of a mixed bag. There's been some successes here and there, which get heralded a lot, and a lot of meh. There's certain kinds of cancer, like multiple myeloma or HPV or chronic myeloid leukemia, where you can look and say prognosis is getting much, much better. Big strides. And then there are others like glioblastoma, pancreatic cancer, ovarian, where there has not been a lot to report on. But we do have a lot of more information at our fingertips now for going after some of these diseases. Maybe I should just start really simple. For those of you who on this panel, you've seen a lot the last 10 years. Just very simply, what would you say would have been the biggest advance, the biggest achievement in cancer of the past 10 years?
In the treatment of cancer?
Yes.
Well, immunotherapy against cancer would be the one that stands out.
Mm-hmm. Now, for those unfamiliar, how would you describe immunotherapy?
Well, to unleash the latent powers of the immune system so that immune cells, which previously were held in check, would now be liberated to be able to aggressively attack cancer cells, which they previously could not do.
There was a disguising mechanism that tumors use to protect themselves from that immune surveillance. And we've learned through pharmaceutical science to interact with those checkpoints and release the brakes, so to speak, so the immune system can attack the tumors. That's been a big one.
It's been a big one because of the dramatic effects it's had on a very small number of malignancies. And like many of the therapies that we may talk about in our session today, it's been dramatically successful in a small number of malignancies, and it's not yet clear whether those successes will be generalizable to a larger number of solid tumors, for example, above and beyond, for example, melanoma and lung cancer. And to the extent that they are more generalizable, how durable will be the response? Because many of the therapies that have been developed over the last decade and a half, 2 decades, are often able to elicit dramatic regression of tumors, only to have these regressed tumors being replaced by relapsing tumors, which somehow acquire resistance to further therapy and now begin to grow aggressively and threaten the life of the cancer patient. So that the victory is only a very transient one, a Pyrrhic victory.
But isn't that part of what's exciting about immunotherapy, in that we are seeing some of these durable, long-lasting responses? Not for everybody, but for 10, 15, sometimes 20% of patients, their tumors are in check for years.
Well, we don't know yet for how long. And by the way, conventional therapies are able to elicit durable responses, indeed cures. So some of the most primitive therapies, radiation, surgery, cytotoxic chemotherapy, when put together, are able to create cures. And those therapies haven't really changed that much over the last decades. In spite of the fact that they're not necessarily molecularly based, they've been ultimately very successful. The dramatic reductions, for example, in the mortality from breast cancer that have been very real have resulted over the last decade in a 35% reduction in age-adjusted mortality from breast cancer. And most of that is due to very traditional treatments, not to immunotherapy. The one molecular aspect of that is that Herceptin, the antibody against breast cancer cells, which is made at Genentech, is one new arrow in the quiver.
Yeah, for about a fourth or a third of breast cancer patients who have that mutation, you can make a targeted drug against that, and that has been very successful.
Yeah.
Napoleon, I'll ask you the same question. What would you say has been the biggest advance of the past 10 years?
I would certainly agree that immunotherapy has been a very important advance. Maybe it's the most visible right now. I think it's perhaps there is some great expectation about immunotherapy. The reality is that, as Bob was saying, that works very well only in a subset of patients. There are major cancer types like a colon cancer. At least the majority of colon cancer do not respond. Glioblastoma do not respond. Breast cancer is very difficult to tell. But certainly in some tumor like in melanoma, there has been people consider it to be a game changer. Even though if you look at the numbers, they're not even so dramatic. Let's say the initial study with anti-CTLA-4 showed that the standard of therapy, which was thought to be very ineffective, at 5 years, 10% of patients were alive. With the immunotherapy, 20%. So still, it's a significant improvement, but still the odds of dying were pretty significant. But what I think has been probably most promising is the possibility to make a number of combination of therapy. There is a very universal combination. One that I kind of found particularly interesting is the recent combination, for example, with anti-angiogenic agent and immunotherapy. Last week, actually, Roche announced that a tumor which has been historically intractable, like hepatocellular carcinoma.
Now, anti-angiogenesis with immunotherapy. So for those unfamiliar, it's one drug that cuts off the blood flow to tumors, and then another one that releases the brakes on the immune system. So you, in theory, have a couple of complementary mechanisms, a couple different ways to attack that tumor.
It's absolutely right. But it can be even more complicated than that, because, for example, VEGF has some unsuspected, unanticipated effect of the immune response. It's been shown now for years, first by Steve Rosenberg. If you give an anti-VEGF antibody, there is a very significant increase in tumor infiltrating lymphocytes in the tumor. The mechanism is not very clear, but the phenomenon has been very reproducible. And this could be some kind of cellular substrate for this additivity, which once again has been seen there is already an approval in non-small cell lung cancer. There's been very exciting data even in renal cell cancer in combination with the Keytruda with the TKI like axitinib. Very substantial. It could become a new standard of therapy. So this is but there is a broad spectrum of a combination which looks very exciting.
We've got targeted therapies, we've got anti-angiogenic, immunotherapy. Michelle, you're thinking about a lot of these different types of modalities, is the word we use. Different small molecules, targeted antibodies, peptides, different ways of approaching targets inside cells, on the surface of cells. What makes you excited about what you've seen the last, say, 5 to 10 years?
Yeah, I think it's all the things that we're talking about. So cell therapies, I would add that. So designer cells, CAR T therapies, for example. So where you take a patient's T cells, you arm them, and then you give them back to the patient, and the patient's own cells kill their cancer. So what's really exciting about all this is how much biology is finally coming to fruition. So in the last 20 years, we understand biology. I mean, these guys are really led the way in this. Really understanding the biology and the individuality of a tumor, what are the different features that all tumors share, needing blood supply, for example, shutting off the immune system, very common. And then what are those things, you describe them as targeted therapies that are very specific to a patient. And so we talk about precision medicine as targeting a drug for that patient, for that disease. And my biggest fear is that every patient will have a unique disease, and we need a unique drug for every single patient. So how can we do that a whole lot faster? But there are also these core things that all tumors share. So linking those two things together has been very effective. And really, it's the underlying biology that makes that possible.
Now, Bob, you were there at the very beginning with RAS. This is a good story, I think, a cautionary tale in some ways about cancer discovery and drug development. It's found in 35% of all cancers. And we've known this for 30 plus years. Nobody's been able to make a drug against this oncogene, this driving mutation. People have known, like, if you could bind with that, shut it down, I mean, that would obviously be hugely helpful and a big seller too. And yet nobody's been able to do it.
Well, it's worthwhile saying that, let's say before 1975, we really didn't know the molecular mechanisms that went awry inside cancer cells. In that year and the next year, here at UCSF in the Varmus Bishop Laboratory, we began to get the first glimmers of the molecular mechanisms. And while in that year we knew almost nothing, in the ensuing decades we accumulated an enormous amount of information on the molecular engines that are driving cancer cell growth. The faith was that if you understand the causative mechanisms of disease, that will lead, as day follows night, to therapies. But the fact of the matter is, understanding causation has not translated in the great majority of cases to truly useful therapies. There have been flashes in the pan. For example, Gleevec has been dramatically successful in treating chronic myelogenous leukemia, and with that success, one began to hope that a whole series of other similar kinase inhibitors, as they're called, would prove equally successful. But the fact is that that's been an idea which has proven illusory, because most kinds of tumors have not proven responsive to similar kinds of drugs like Gleevec, and to the extent they have, it's only been for 6, 8, 10 months with no overall gain necessarily in long-term longevity of the cancer patient. So it's been very frustrating. I thought that if, in 1982, you could begin to develop some general laws about how cancer cells arose. And the deeper we all, that is the community of cancer researchers, went into this, the more we realized that cancer is a much more wily and elusive foe than any one of us would have suspected. There's probably 200, 220 different subtypes of cancer. Even within a given subtype, every patient's tumor is different. And most frustratingly, these tumors are very plastic. They can change like chameleons from one biological state to another and thereby elude therapy.
The new tools that we hear a lot about for gathering information about cancer, like next generation genome sequencing, biomarkers, and sophisticated high resolution imaging, flow cytometry, cell counters. I mean, we've got tremendous tools to look deeper and deeper into the cell.
Generating data, but not necessarily generating insights into what's going on.
Yeah.
So there's a love for gathering enormous data sets, sequencing thousands of cancer cell genomes, with the presumption that somehow this will afford insights into new ways of treating patients. But I'm become a little bit jaundiced about that, a little jaded.
It has redefined our understanding of cancer. I mean, we don't talk about it in an organ-based way. We don't just say breast cancer. We say HER2-positive breast cancer, or HER2-negative, or triple-negative breast cancer. And there's all kinds of, you mentioned 220 different subtypes of cancer. I mean, we could have this conversation a couple years from now, and there might be 300. I mean, we keep slicing it, and as Michelle said, everybody's unique in a certain way. Michelle, are you a little more optimistic about these tools? Are we just kind of struggling to figure out what to do with all this data, and we'll get there?
Yes, we're struggling to figure out what to do with all this data, which doesn't mean the data won't ultimately be useful, but we need to be able to make drugs faster as well. So the more wily the disease is, the more we're playing whack-a-mole. So that as soon as one subpopulation of the cancer goes down, another subpopulation arises and then gives rise to resistance, which is why you can often have a gap in, so you can increase the amount of time before somebody progresses on disease and yet not improve their overall survival because the disease comes back really aggressively. So we have a lot of complexity there, which doesn't mean that all that data won't eventually help us. I think part of it is that, so RAS is a good example. We understood that it was important biology, the nuances of it we're still trying to understand, but also targeting RAS itself, it was considered an undruggable target. We knew it was there, we knew it was important, but we didn't know how to get a molecule to stick to it in a way that would block its function. And so because of that, we go after things that are a little bit away from it, that we do know how to target, and they're a little bit distal from the thing that we're really interested in. And those work for a time, but since they're not the core, they're the periphery, then maybe that's a reason we can generate resistance. Whereas now we're starting to target subclasses of RAS, and also the RAS protein-protein interactions that it makes. So RAS functions by being turned on all the time instead of being on and off. And when it's turned on all the time, it signals for the next baton to be passed, and that eventually leads to cancer growth, to uncontrolled growth. So if you can block its ability to pass the baton, and you can figure out technically how to do that, that's been a big breakthrough in looking at the problem a little bit differently.
Has there been a problem with a little too much narrow reductionism in biology? Because we know from so much of your work over the years, Bob, that there's oncogenes, there's tumor suppressor genes, and they're often achieving some kind of balance when we are normally going about without cancer. And then something happens where that balance gets thrown off, allowing this uncontrolled cellular growth, which is cancer, to occur. Do we need to throw more at it? Like 2, 3, 4 different combo agents in many cases to get where we want?
That will surely be the long-term solution. Napo is very specialized in that. But we have to remember that when you throw 2 agents at a cancer, there's also a lot of rate-limiting toxicities. You might end up killing the patient before you kill the tumor, which is always awkward for a clinical oncologist.
Not the goal.
These large data sets you were talking about before, I'm not at all convinced they will accrue to our benefit. People who are gathering them said, well, we'll just use artificial intelligence to interpret what's going on there. I must say, I often say, after you've talked with somebody who's a proponent of artificial intelligence, if you shake hands with that person, you should count your fingers afterwards, because I don't necessarily totally believe all the claims of those people. I fear we're going to have large data sets that will be archived somewhere and will remain forever uninterpretable in the sense of not yielding significant specific insights into how to treat patients, tumors.
I still say a few words about,
Yeah, it's on.
Just hold it close.
Combination, even the HER2 kind of axis, there's there is a totally unexpected, counterintuitive benefit to combine the 2 agents that target the same pathway, like trastuzumab and pertuzumab, okay, interacting with different epitope.
These are 2 different antibodies from Genentech that go after the HER2 protein that's overexpressed in breast cancer. So it's like a double blockade of the same signaling pathway for tumor growth.
It's counterintuitive, because most of the time, if you block the same pathway with another agent, nothing happens. You, as Bob will say, you're only increasing toxicity. In this case, there has been a very significant improvement in survival. So it is very difficult to predict at times.
It's easy to throw stones. In other words, it's easy to be curmudgeonly, as I'm being, to be a negativist. Because if we were against everything and we were skeptical, we'd still be in the Stone Age. But one has to admit the fact that it's been very frustrating in terms of converting basic insights into the etiology, into the causation of disease, into truly useful therapies. And by the way, if I could throw another hand grenade,
Please do.
if you look at successes in reducing cancer mortality, some of them will come from new drugs, which, as you know, cost often $750 to $1 billion to develop. We're going to run out of money for developing all these new drugs. And to the extent that we get all excited about improvements in cancer therapy, to the extent that they succeed, major advances are often reducing cancer mortality by a factor of 0.5%, or let's say a proportion of 0.5%. And soon, with the aging of the population and the increase in the age-related disease of cancer, and actually Alzheimer's, we're just going to run out of money to treat all these cancers aggressively, because the number of cancer patients is going to increase progressively because of the aging of the population. But the productivity of the economy is not going to increase concomitantly. And so I'm increasingly of the mind that the major advances in reducing mortality can come only and will come only from reducing incidence, which is not very popular among my colleagues who have devoted their careers to developing new kinds of therapy.
Anti-smoking campaigns, for instance.
It's the messy business of human psychology and behavioral modification, and none of us wants to become involved in that, even though the benefits potentially are orders of magnitude more than anything we in the basic cancer research community could think of achieving.
I actually agree that I think the pricing and access issue around new cancer therapies is going to be a really tough nut to crack. Some of these new cell therapies that Michelle alluded to cost $375,000, $475,000, and they work very well for a small group of people. But if you were able to generalize that across larger numbers of cancer patients,
scalability,
the math is just impossible. Who's going to pay for that? And how are people going to be able to get it? It's a real problem. So, but Michelle, there was something, he made a remark about artificial intelligence and large data sets in a very skeptical way. You're not of that same mind. Why?
Well, I mean, what he's talking about is the bioinformatics is figuring out what to target about a cancer based on data. What we're working on, I'm part of a consortium called ATOM that's working with several national laboratories, big data and drug discovery experts, National Cancer Institute, to use artificial intelligence to more rapidly develop drugs. So it's more at the okay, once you've identified a target, that's the big problem that we've been talking about really is what to target. But maybe you don't have to be as smart about it if you can target it faster and get to a proof of concept faster. So get a molecule that you can test faster and more efficiently. One of the big problems, so using artificial intelligence to really speed up our design, make test cycle. So drug discovery is it's a cross between science and engineering. So we have to design a drug based on what we know about how the structure leads to its function, the structure of the molecule leads to the function of the molecule, make it work better. And usually what we do is we first make the molecule bind really tightly to the thing we want it to bind to, and then we try to make it survive in the liver so it doesn't get seen as food or poison and get gotten rid of, so it gets to the tumor. And then we try to make it safe so that it kills the tumor before it kills the patient. And if we could do those things in parallel instead of doing them linearly, we could speed up the process a lot. And we've seen this in the history of drug discovery, moving some of those later stage issues earlier in the discovery process, and it's really changed why drugs fail. But the key issue for, cost is a key issue. Key issue is failure. So 1 in 10 drug projects that we start in the pharmaceutical industry make it to the clinic, and then 1 out of 10 that we start in the clinic that we put in a patient makes it to a successful drug.
Huge amount of waste in the system. The head of Merck Research Labs, Roger Perlmutter, famously said, whenever a drug gets approved, it's a bloody miracle.
Yeah.
Despite all this data and predictive power we think we have.
That's right, because it isn't really an engineering problem because we don't understand biology well enough, right? We can't map out what it needs to look like we can some other complicated thing, like my colleague Adam likes to talk about building an airplane. What's the difference between building an airplane and building a drug? we understand the physics of airplanes and we don't understand the physics, the biology of drugs. So if we can, anything we can do to do that faster, to do that more efficiently, that's one of the things about antibody drug discovery that's been so impressive, is that you understand the biology you're targeting, the molecule's easier to make, you can get to a fail or succeed process point faster.
But an antibody only binds with targets on the surface of cells, and that's a pretty small number of targets, really, compared to what's inside the cell. And that's where small molecules, like your traditional aspirin, they can get inside cells.
Every modality has its time and place, right? And so I used to work at a company in the early days of small molecule discovery for protein-protein interactions like VEGF binding to its receptor. And there we said, well, if we can make a molecule that does what an antibody does, it would blow the antibody out of the water. It's so much easier to make. It's so much cheaper. But actually, antibodies are very good drugs. They have some very good properties that small molecules. So what's the right modality for that target? What's the right way of looking at the problem? And if we can do that faster, then we can fail less or we can fail faster and move on.
But there is a problem which I think you are alluding to, the predictability. The fact, that the animal model is so poorly predicted. This is a very, very serious problem. We don't fully understand, why this happened.
If you've got to get cancer, it's good to be a mouse.
Yeah, exactly.
We've cured cancer in mice many, many times.
Well, it's really interesting, too, because why did it take us so long to come around to immunotherapy? Because we treated our drugs in immunocompromised mice so we could stick a human tumor in the mouse. So that's one thing that really holds us back, those assumptions we don't even realize we're making that make it easier to kill a xenografted tumor in a mouse than it does a human tumor in a human.
And 10 years ago, nobody really believed, except with very few exceptions, in immunotherapy. That was just not mainstream biology 10 years ago.
It was smoke and mirrors, and it's not that now.
Yeah, it's front and center.
Yep.
Well, when I was a postdoc, the first postdoc at Genentech was the year Herceptin got approved, which dates me quite precisely. And and then nobody thought antibodies. For a long time, we were swimming upstream with antibody therapies, and now they're mainstays for a lot of diseases. So just have to be the one who's pushing forward and following the data instead of the...
Are the tools, what excites you about the tools on your workbench these days, Michelle? I mean, is there something about the way we can make small molecules so that they can bind in certain ways with certain kinds of protein-protein interaction targets? I mean, the analogy sometimes people use is sort of like these two kind of like, vertical rock faces that don't really have much of a toehold on them, right? But now we're figuring out ways to make molecules that can sort of scaffold or lattice their way in there.
Right, so some of it is, some of it is figuring out what's the right target, what's the right technology to use to find molecules that bind that target, because you get what you screen for, is the adage. So you have to use the right set of molecules in the first place, and this is maybe a place where virtual screening or where artificial intelligence can help, or different types of experiments, different types of libraries. So it's really matching the technology to the problem, the binding problem in the case you're describing. And then also understanding that most proteins are not really these two cliff faces coming together, but they move. And when they move, sometimes they have pockets that you can stick a molecule into. So how do you find those? How do you set up your system to find those rare events? And then the next thing is, when is a small molecule just not the right way to go? But there's some other modality, some emerging modality, nucleic acids or something more next gen, that's going to be the better way to attack that particular problem.
CRISPR, we hear about RNA interference. There are lots of different ways to interact with targets now that we didn't have 10, 15 years ago.
I think the current model, what we're doing, I liken it to a big ocean liner about to hit a large iceberg, given the economic realities of therapy, drug development, therapy development. And the question is what my colleagues are doing here, is it going to rescue us before we hit the iceberg? Because the era of developing $750 million drugs, which is what they cost now, it's almost over. Nobody will be able to afford it anymore. What are these people smoking who think that we can?
Well, so where do you think we're going to end up here 10 years from now, Bob? Are we going to have a flat line here on cancer death rates and 5-year survival rates? Are we going to get nowhere?
I honestly don't know. I don't mean to be so negative. Again, if we were all negative like I'm being, we would still be in the Stone Age. But the forward trajectory over the next decade, it's not totally obvious to me, given these increasingly acute economic realities.
So one thing that you see when you look at pediatric ALL, for example.
Acute lymphoblastic leukemia.
Pediatric leukemia, yes. It's a rare disease, and it used to be nobody survived, and now most kids survive, and they're old drugs. They're, from a standpoint of biology, boring old drugs, but decades of exactly how to use it, exactly how do you dose it, how do you combine things. So you see incremental improvements in cancer over time, and then you try to layer these breakthrough drugs on top of that incremental improvement in how to use drugs. So we have this incorrect idea that as soon as a drug is approved, it's done, it's on the bookshelf. It's not. It's a living thing that you're always trying to improve the patient population, the timing, the dosing, the combinations.
Actually, we learn a lot about mechanisms years after a drug has been on the market. Oh, it actually works in another way in addition to the one originally theorized.
Right. Yeah.
It throws forth new hypotheses.
Yeah, you can be a little too smart if your drug is too specific.
Napoleon, do you think we're going to bend the curve on survival in the next 5 to 10 years?
I don't know if this is kind of the prediction, which are very dangerous. There are some famous people, famously difficult to. But I think I'm not so pessimistic. I think that there has been some. If you look, for example, survival in colon cancer from the days with the 5-year fuel on, with the multiple agent, there has been very significant, meaningful improvement, and I suspect we'll continue to see that. Maybe we won't see this quote unquote a cure for cancer, but it'll be meaningful, improvement in survival, in progression-free survival, etc.
Maybe AI will help us with the combinatorial problem of like how do you take 2 or 3 agents together, at what dose and what schedule? I mean, there's a lot of possibilities there.
Just count your fingers afterwards.
Okay, I'll remember that. And I think with that, I think we're out of time. Thank you, everybody, for listening.